This video will cover the anatomy and physiology of cells, corresponding to Chapter 3. The cell is the basic unit of structure and function in living things. Human cells, varying in size and shape, are typically microscopic. The human body is composed of about 100 trillion cells. A cell is the fundamental living unit of any organism. It's crucial for producing energy through metabolism. Cells can mutate due to accidental changes in their DNA. Cytology is the study of cell structure and functions. This diagram shows a sectional view of a cell, illustrating various organelles and components such as the nucleus, cytoplasm, plasma membrane, and different types of filaments and vesicles. The cell or plasma membrane is a thin, dynamic membrane that encloses the cell and controls substance passage. It is described by the fluid mosaic model, composed of a phospholipid bilayer with dispersed protein molecules. In the fluid mosaic model, the membrane surfaces are hydrophilic due to polar phosphate heads, while the internal portion is hydrophobic due to non-polar fatty acid tails. Membrane proteins also possess both hydrophilic and hydrophobic regions. This visual representation details the plasma membrane structure, highlighting the lipid bilayer, phospholipids with hydrophilic heads and hydrophobic tails, cholesterol, and various integral and peripheral proteins like channel proteins and glycoproteins. The plasma membrane is formed by chemical attractions that hold the phospholipid bilayer together, featuring hydrophilic phosphate heads on the exterior and hydrophobic fatty acid tails internally. The plasma membrane serves as a boundary and acts as markers to identify cells. It plays a significant role in transportation, and cell recognition proteins allow cells to recognize others. Membrane proteins include glycoproteins, which act as identification markers. Other membrane proteins are receptors that react to specific chemicals, permitting a process called signal transduction. The cytoplasm is a gel-like matrix containing water, enzymes, nutrients, wastes, and gases, encompassing the organelles. The fluid part is called cytosol. Most metabolic reactions occur in the cytoplasm. The endoplasmic reticulum is a network of interconnected parallel membranes continuous with the nuclear membrane. The rough ER is studded with ribosomes and is involved in protein synthesis and transport. The smooth ER lacks ribosomes and is vital for lipid and cholesterol synthesis and calcium storage. This image illustrates the rough endoplasmic reticulum, RER, showing its close proximity and continuity with the nuclear envelope and the presence of ribosomes on its surface, distinguishing it from the smooth ER. Ribosomes, thousands of which are in every cell and many are attached to the RER, are non-membranous structures made of large and small subunits composed of rRNA. Their primary function is protein synthesis. Proteins released from the ER require further processing in the Golgi complex. The Golgi apparatus consists of flattened membranous sacs called cisternae, arranged in stacks. Its main function is the modification, packaging, and transport of proteins, and it encloses digestive enzymes to form lysosomes. Lysosomes are spherical membranous sacs containing digestive enzymes. They are often called suicide sacs because they destroy things the cell no longer needs. Autolysis is the process of worn cell part digestion by autophagy. Peroxisomes are membranous sacs containing oxidase enzymes. Their function is the detoxification of harmful or toxic substances, such as alcohol and formaldehyde. They also convert hydrogen peroxide into water. Mitochondria are kidney-shaped organelles. The inner membrane is folded into shelf-like partitions called cristae. Mitochondria are the powerhouse of the cell, where cellular respiration releases energy from glucose. The nucleus, the cell's central core or control center, is the largest organelle, filled with nucleoplasm. The nuclear membrane is a double membrane with nuclear pores, which allow the passage of substances like mRNA and ribosomes. Nucleoli are spherical bodies within the nucleus, composed of RNA and proteins. Their essential function is the synthesis of ribosomes. The cytoskeleton is a network of fibers throughout the cytoplasm. It provides support to organelles like the endoplasmic reticulum and mitochondria. It includes structures like microtubules and microfilaments. The cytoskeleton offers mechanical support and is involved in cell motility, utilizing motor proteins. These rod-like pieces provide support and enable movement within the cell or of the cell itself. Microfilaments are solid rods of globular proteins, a key component of the cytoskeleton offering structural support. They can slide past each other, which causes cell shortening. Intermediate filaments are twisted protein strands, slightly thicker than microfilaments. They form a significant portion of the supporting framework in many cell types. Microtubules shape the cell, guide the movement of organelles, and help separate chromosomes during cell division. The centrosome, near the nucleus, is the microtubule organizing center. It coordinates microtubule assembly and breakdown and plays a role in cell division. It contains a pair of centrioles. Centrioles are self-replicating structures composed of bundles of microtubules. They are critical for organizing cell division. Cell membrane surface modifications include cilia, which are short, hair-like extensions, aiding substance movement, and flagella, a tail-like projection for cell locomotion, like in sperm cells. Microvilli increase surface area, found in the digestive tract lining. Membrane junctions include tight junctions, which are impermeable and prevent leakage, like in the blood-brain barrier. Desmosomes are anchoring junctions that prevent tissue fraying, and gap junctions allow the passage of chemical substances between cells, as seen in the heart. Tight junctions seal the space between adjacent cells, preventing leakage. Desmosomes act as spot welds, holding cells together and distributing tension across tissue. Gap junctions are channels between cells, permitting the rapid passage of small molecules and ions. This is important for communication and coordinated function, such as in heart muscle contraction. Transport across the plasma membrane occurs through two main types: passive transport and active transport. Passive transport requires no ATP, moving substances from high to low concentration. Examples include simple diffusion, osmosis, facilitated diffusion, and filtration. Simple diffusion is the random mixing of particles in a solution. Substances move down their concentration gradient until equilibrium is reached, meaning particles are evenly distributed. This diagram illustrates the process of simple diffusion, where molecules move spontaneously from an area of higher concentration to an area of lower concentration across the lipid bilayer. Facilitated diffusion involves movement through channel proteins or transport proteins. This allows small inorganic ions like Na+, K+, and Ca2+, as well as glucose and water-soluble vitamins to pass. The rate depends on the number of available transporters. This illustration shows the various mechanisms of diffusion across the plasma membrane, including simple diffusion through the lipid bilayer and facilitated diffusion via channel proteins or carrier proteins. Osmosis is a passive process involving the diffusion of water across a selectively permeable membrane. Water moves from a high concentration of water to a lower concentration of water, which means from low solute concentration to high solute concentration. Membrane permeability significantly affects both diffusion and osmosis. The plasma membrane structure, including the presence of channels and carriers, determines which substances can pass and how water moves during osmosis. Tonicity describes how a solution affects cell volume. A hypertonic solution causes cells to shrink or crenate. A hypotonic solution causes cells to swell and hemolyze. An isotonic solution maintains cell size. This visual demonstrates the effects of hypertonic, isotonic, and hypotonic solutions on blood cells, showing how the movement of water due to osmotic pressure alters cell shape. Active transport moves substances against the concentration gradient, from lower to higher concentration, requiring a carrier and energy. Endocytosis is a type of active transport that brings substances into the cells. Active transport involves solutes moving against their concentration gradient using carrier proteins. This process is driven by ATP or energy stored in ionic concentrations. Types include primary active transport, secondary active transport, endocytosis, exocytosis, and transcytosis. Primary active transport uses ATP and transporter proteins, a prime example being the sodium-potassium pump, which moves ions against their concentration gradients to maintain cell membrane potential. Transport in vesicles, known as endocytosis, is a form of active transport used for large particles across the plasma membrane. Key types include phagocytosis and pinocytosis.